Alright. For the last two blocks, when we've been starting new blocks, I've been kind of like glossing over the ELOs. For parasitology, I think it probably warrants a quick touch up on what we really need to focus on in parasitology. And I think that'll set up a little bit more of the reasons why we get into such detail about life cycles, parasitic life cycles in particular, when it seems like they're so detail laden. The reason why is you really don't understand how to prevent acquisition of these diseases or treat them unless you have a good understanding of the life cycles. So, but today we'll mostly start, this is just an intro step, so we'll talk about terms that are used to communicate and understand parasitology. It's like its own set of vocabulary. We will be building on things we've learned in other blocks as well, but we'll spend some time with that. And then Monday is when you'll start seeing like how detailed these life cycles can be with parasites. Today we'll also, actually I said on the table list, but when we start off in the lab ID section, that'll go over some of the specific lab tests we do for parasite testing, like the OMP test, if you've heard about those. We'll talk about those a little bit, some other tests, fit smears. And a very little bit about collecting and handling specified parasitological specimens, mainly like stool specimens, but others as well. Okay. So today, just broad stuff. We just got done with a block that was mostly about virology, right? Like 70% virology, but we also talked about some eukaryotes. We talked about mycology, and we talked about some prokaryotes, some acid fast bacteria. So we are in a world of clinical parasitology. We're back in eukaryotic land. Not talking about bacteria anymore. We're not talking about archaea. We're talking about worms, which are animals. Helminths is the technical term. And then we're talking about these unicellular eukaryotes called protozoans. And then that last lecture we'll have will be on the ectoparasites that live on the outside of our bodies, as opposed to endoparasites. As a field, out of everything we've talked about so far, the major disciplines of microbiology, when we talked about bacteriology, when we talked about virology, mycology, this is one of the most ignored branches of microbiology, especially in the United States, where we have really good, pretty well developed hygiene systems, good public health responses, good education about disease transmission. But we still have lots of parasitology here, so you'll see parasitology for sure once you get out there and see patients. So in the United States, we certainly have endemic parasitology diseases, and if you get out and travel, we'll talk about some of those specific parasitic diseases that are associated with certain endemic regions that might affect traveling populations. Refugees and immigrants are another population that can be particularly susceptible to parasitic diseases, immunocompromised individuals, military populations. In the US, like I mentioned, we do have it pretty good, but some of these diseases, if you look down here at some diseases that are endemic in the United States, particularly these that I put on this list over here, are pretty well known. You guys ever heard of pinworm infection? Enterobiasis? Yeah, so that's worldwide. We certainly have that in the US. We'll be talking about that. Toxoplasmosis. Heard about toxo? It's extremely common. One of the most common parasitic diseases worldwide. What about trichomoniasis? What? I don't know what that means. Oh, you're thinking of trichinella, trichinosis. Trichomoniasis. I was like, bears. This is an STI.
Also look out some whoa.
Yeah, I mean, I'm sure bears have their own STIs, but this is ours. Trichomonas vaginalis. So it's the only like hardcore legitimate parasitic STI that we're covering this course. Yeah, Aaron.
Have you ever heard of, like, it's a thing, maybe, like how historically people from the South were viewed as like dumber?
Yeah.
And then there was like the correlation between like footform because they went around barefoot.
Yeah.
Is there validity to that?
There's total validity. There's a whole PBS documentary. There's a book
You
written
watch
about
your
it.
mouth.
They're talking about making a movie out of that story with Matthew McConaughey playing one of the lead sellers. talk about, rheumatoid infections, talk about hookworm and Decatur americanus, which was like endemic in the south, down here. You know, southerners running around barefooted near the trains and hookworms penetrating in their feet. Like open, what you call them, like outhouses. Anyway, yeah, we'll talk all about that. But that particular disease, hookworm, causes like a gradual leaching of nutrients and anemia. So particularly in kids who are infected with hookworm, it causes stunting, delayed mental development. Even in adults, the anemia has this kind of like stunting effect on people. So southern laziness was a characterized thing, and people think that it was attributed to hookworm infections that were just rampant at the time.
It didn't carry over, did it? Like it's done now?
No, it's gone. It's still here. I mean, they've done surveillance studies in the soil, and yeah, it's wherever human feces are. Oftentimes, they're not.
But it doesn't linger through generations.
No. It's not eradicated. It's not eradicated. We still get cases of hookworm in the south, just not like we used to in the antebellum, post-Civil War era.
Okay, excellent.
So, yeah, next one. Real quick, geodiasis, you guys heard of geodiasis? You already did? Okay, so that's like all over the country, but particularly that's kind of like probably most endemic in the Pacific Northwest. We'll talk about that. That's a major diarrheal disease. Cryptosporidiosis, that has an association with immunocompromised folks, but it's also associated with like huge outbreaks, waterborne outbreaks. One of the biggest outbreaks in US history was when the city of Milwaukee, right here, had a cryptosporidiosis outbreak. And they had some sewage leak into their water supply. And I don't have the numbers fresh in my head, but a lot of people got cryptosporidiosis because it got into the city water supply. That was in 1993, I think. That's a big outbreak there. And then Chagas disease. Anybody heard of Chagas disease? A couple people? Yeah. So this is American trypanosomiasis. You've probably heard of African trypanosomiasis, African sleeping sickness. But we have our own type here in the southern region, like where we are now. We get cases of Chagas disease. So we'll talk about that a little bit too. Lots of stuff. In terms of the toll on the human population, malaria is one of the big three. We talked about the others, tuberculosis and HIV. So malaria is right up there. These estimates might be, they're pretty high. I've seen lower sources, and we are making progress in reducing the number of cases of malaria and the number of deaths. But this is still probably the highest burden due to parasitic infection worldwide, malaria cases. 2.5 million deaths annually. A good chunk of those deaths are in children. So it's still remains a problem, something we've been spending a lot of money and investing a lot of resources in for a very, very long time. Lymphatic filariasis, have you heard of filariasis before? Yeah, causes elephantiasis. It's not really high mortality, but it is significant morbidity. It causes disfigurements, swelling of the limbs. Leishmaniasis, that can be a fatal disease. Not a huge death toll annually, but certainly can be fatal. Hookworm infections, not a lot of fatalities associated with it, but again, significant morbidity, not mortality. And this number is definitely reduced more in the developing world. 400 million people are susceptible to infections. 400 million people. Schistosomiasis, if you've not heard of this is big in Africa and Southeast Asia. 290 million people globally have schistosomiasis infections. 5,500 deaths per year. Trichuriasis, 400 million, and so on and so forth. But Chagas disease, 9.4 million. It's a significant number of deaths due to some of these parasitic diseases. All right, getting into terms a little bit. The first two, an obligate parasite or a facultative parasite, we're familiar with these terms from talking about bacteriology. When we talked about something that's obligate, what do you think it means in terms of its relationship to a host, like a human? You gotta have it, right? Whereas a facultative parasite, something like this guy in the upper left, this is Naegleria fowleri. It's a protozoan facultative parasite. What'd you say, John?
That radiating, yeah,
brain-eating amoeba. So it's just minding its own business in ponds, in water, right? It doesn't need to infect humans at all. It feeds off of like algae or whatever its food source is out in these ponds. Where we come into contact with it is when we go swimming in these places with Naegleria fowleri, and it gets forced up our nose and then causes this very rapidly fatal disease called primary amoebic encephalitis, the brain amoeba. But it's a facultative parasite. Most of the ones we're talking about are more obligate parasites. I think they need a host. Two terms that I won't use very often, but it's good to at least have a handle on what they mean. Homoxenous parasites versus heteroxenous parasites. Most of the ones we're going to be talking about will need multiple hosts, like malaria, Plasmodium species, the cause of Asian pandemic, needs mosquitoes and humans. That pinworm that I mentioned, Enterobius trabecularius, that just needs human, that's all it needs. Head lice, one host, just humans, that's it. So those are homoxenous parasites. And then the last two, parasites and ectoparasites. You have something like this giant intestinal worm, Ascaris lumbricoides. That is an endoparasite. It's not going to be hanging out on the exterior of your body like a mite would, or a tick, or fleas, lice, any of the ectoparasites. So pretty intuitive stuff there. Yeah, this is something that was originally in your deck. I took it out, but you'll see more pictures of this. If you had to guess whether or not this particular parasite was an obligate parasite or a facultative parasite. I think obligate, yeah. It looks like it's designed to do some damage. Isn't it? Yeah. So those are chitinous teeth, and they do exactly what they look like they do. They're used for attachment and latching onto intestinal tissue. And they do cause injury to the colon when they do that. Talk about those more later. Alright, we talked about types of parasites. Let's talk about types of hosts now. We've got the reservoir hosts, which we sort of talked about a little bit last block, especially with viruses. For example, when we were talking about West Nile virus. Because remember, another animal that might be considered a reservoir species for West Nile. Birds, excellent, yeah. Yeah, crows, blue jays, yeah, so those are reservoir species for West Nile virus. When we were talking about leprosy, Mycobacterium leprae, one of the reservoir species for that was the nine-banded armadillo. It's a considerable reservoir for that bacteria to hang out in. For parasites, we talk about this much more frequently. Doesn't necessarily cause a lot of damage to the host, but it's like a sink for that parasite to have continued presence. So the example here is with Toxoplasma gondii, the parasite that causes toxoplasmosis. That spends a lot of time in rodents. That's its reservoir species. Rodents get eaten by a lot of cats, so they transmit it to the cats. But the rodents themselves don't really suffer too much when they're infected with Toxoplasma gondii. They make a good reservoir species. The definitive host is, by definition, the host that harbors the adult or sexually reproducing stage of the parasite. Especially early on, the first couple of parasites we talk about, I will stress which one is the definitive host and get used to sort of recognizing what that means. But for example, on this life cycle, this is showing the life cycle for Schistosomiasis. So, like a non-human life cycle here. In this particular life cycle, we have a non-human definitive host, just a bird that hangs out in the water. It gets infected, and then as it's infected, it'll pass an egg. That egg gets released into water, develops into the next stage, which is Miracidium. That stage is able to infect a primary intermediate host, which is the host harbors the immature, asexually reproducing forms. It develops in that snail host, exits the snail, goes back into the water with its tail as a cercaria. It can swim around and find another host, another bird, or it can be taken up by a secondary intermediate host. If nothing eats the intermediate host, that transmission cycle is broken. But let's say this heron, or whatever this bird is, parasites have really complex life cycles. They might involve one intermediate host, two intermediate hosts, sometimes three intermediate hosts. It gets pretty complex. So that's what an intermediate host is. Harbors that asexually reproducing form of the parasite, but it's required for development. When we talk about incidental or accidental hosts, this is a host that is not normally used for development. So say you've got your Cercaria swimming out in the water there, and it would be infected for this bird. Instead of the bird, it sees a human splashing around, and it penetrates into the human and tries to establish infection there. If it's not human adapted, it would probably fail to develop further in that host. So that would be an accidental or an incidental host, didn't intend to infect that. Those are usually, but not always, dead-end hosts, which means that that host is inappropriate for further development of that parasite. Lots of examples of that. Basically, like, for example, if we get a tapeworm, I'll table that example. It's kind of a convoluted example, but dead-end hosts, that's something else that I will point out a lot, especially when we talk about tapeworms. And this example is a good one, it's really worth talking about. You guys heard of the dog heartworm? Dirofilaria immitis. So that, in dogs, when this particular parasite infects dogs, it will get into their heart and it will be able to develop in their heart. In humans, it will go into the heart as well, but it doesn't receive the right environmental signals to develop. So it basically gets pumped out to the lung fields and it dies in the lungs. Dead-end host, or not the right host for Dirofilaria immitis. In terms of transmission, parasitic transmission, I've had a couple different terms here to be familiar with. The life cycle itself talks about the entire life cycle. An example on this slide is a little horrifying, but this is the entire life cycle for Plasmodium. And we will go through this, it's not in your deck, but we'll go through it when we talk about malaria and Plasmodium. So that's the entire life cycle. May require one host to complete the life cycle. What term would that describe? More than one host? Heteroxidous, yeah, multiple hosts. So it can be pretty complex. The infective stage, developmental stage in which a parasite is capable of invading a host. This is important. For the example of hookworm, when you're walking along barefoot in the grass, just because we've talked about it a little bit already, what is actually infective is a little larvae stage. That larvae is equipped to penetrate into your skin. If you encounter it before it hatches and becomes a larvae, it's not in the right infective stage and it cannot infect you. So that's important to larvae. Infective route, is it ingested? Is it like Cryptosporidium, where you ingest contaminated water? Skin penetration, like that hookworm. A vector bite or a mosquito carrying Plasmodium, transmitted malaria. Congenital transmission. Anybody know of any congenitally transmitted parasitic diseases? Toxoplasma, toxoplasmosis. Warn pregnant women, be careful about handling your cat, about cleaning out your kitty litter. So congenital transmission. Infective mode, mechanism by which the parasite invades the host. Active mode or passive mode? Pretty straightforward stuff. Passively, you're just, coming across these parasites almost accidentally, ingesting these Giardia-cysts. It could be you, it could be a muskrat, it could be a beaver, ingesting these cysts from contaminated water. With an active mode, the mosquito isn't so much doing it themselves, it's just taking a blood meal, but it still has to feed, right? So that's an active process, an active injection of this parasite into your bloodstream. Vectors are the same as what we've been talking about with last block as well. Vectors being like mosquitoes, sandflies, mites, fleas, and autoinfection. This is a new term and it's very important in this block. Reinfection, in which the host becomes its own source of infection from a parasite already present in the body. There's only 2 or 3 different parasites that we're going to talk about in this block that are capable of doing autoinfections. One of them is the pinworm, Aerobius furnicularis. Strongyloides is another, Hymenopus nana is another, so we'll talk about all those, but the reason why those are so important is because when you're your own source of infection, you can get hyperinfection. If you can't break that transmission chain, you just keep infecting yourself, you get a heavy parasite burden. This might be a little overwhelming right now, but let's just see a couple questions. Can you read that text there? Above the mosquito, that guy's shoulder. Injects what? Sporozoites, yeah. So spore, tough, zoids, animals, tough little animals. Injection of sporozoites. If I asked you what the, let's say, effective route was for the diagram to this life cycle, effective route for humans, what would you say? Yeah, so there's a couple, I think, correct answers, right answers. The skin penetration, direct injection through a vector, through a mosquito, transmitting it to the blood. But not fecal oral, not sexual, not congenital, right? This is direct penetration through a biting insect. Then we have a bunch of stuff that we haven't talked about yet, but basically asexual development in the human host. So this is an intermediate or definitive host if the asexual reproduction cycles are happening. If the asexual forms are happening, does that make the human an intermediate host or the definitive host? Intermediate. Exactly. And then down here we have now development to a stage where we have male and female gametocytes that get picked up by the mosquito and then they fuse, which makes the mosquito the definitive host, their intermediate host. The definitive, yep. The definitive host is the one where sexual reproduction occurs. So it's a little bit counterintuitive, but that is the definitive host for Plasmodium species. So if that's super overwhelming, that's what this entire block is covering in minute detail. A couple of the quick things here. The diagnostic stage is helpful for us because this is a stage where we look at a specimen from a patient who's exhibiting signs and symptoms and actually detect that organism. When we look at malaria, we will draw blood from that patient and we'll do thin and thick smears. And if they're at that diagnostic stage, we can actually identify what type of malaria, what type of Plasmodium species is infecting that patient. So that's important. But we'll go over that in more detail later. All right, factors associated with parasite pathogenicity. One is the infected dose and exposure. So are you, especially with fecal oral route, you're ingesting a huge amount of this parasite versus a small amount that will have an effect on the disease course. Exposure, if you can get away from the transmission source, that's great. A lot of people can't though because they don't have sanitary hygienic conditions. So their exposure is continued and that makes it almost impossible for them to really remove themselves from the parasite. A lot of these different parasites will have different ways of attaching or adhering to the host. Some of those unicellular protozoans like Plasmodium, it affects red blood cells, erythrocytes, right? So it's able to adhere to those red blood cells. Giardia, right here, this left scanning electron micrograph. This is another protozoal infection. It's an intestinal protozoan. So depending on your infective dose and your ability to clear it, you could get a really heavy burden of Giardia in your gut, right? So heavy that if you look at the way these guys are basically lining or blanketing the intestinal tract, there's significant pathology there. What do you think happens when you have your entire intestinal tract covered and blanketed by this parasite? Yeah, you're running malabsorption disorders, basically. So the diarrhea that results from giardiasis is distinctive because of that malabsorption that's caused by heavy infections like this. These things reproduce by binary fission, and if conditions are good, they just keep reproducing. Heavy burden. These are examples of protozoans that can attach. Even the big worms, like tapeworms, for example, they have to attach too, otherwise they would be let loose out of the GI tract. So that's attached to your mucosal epithelial intestinal parasites like hookworms and tapeworms, they're using the same nutrients that you are. So some of them will some of them will feed directly from your colonic tissue and draw small amounts of blood from the intestines. Other will just absorb nutrients through their tegument, through their skin basically. So they're doing minimal damage to you, they're just siphoning away calories and nutrients from you as you're eating things. Even something like malaria, when it's infecting your red blood cells, it's causing slicing red blood cells. When you have a really severe high level of parasitemia, bad malaria infection, you start becoming anemic. Because what happens when you have red blood cells that are lysing uncontrolled like that? You lose your oxygen carrying capability with your red blood cells, so you can become anemic from that, and your iron stores as well. Toxin production and tissue damage. Some of these organisms will secrete degradative enzymes, like schistosomes, when they're migrating around tissues. The liver, for example, they're aided by degradative enzymes. Blockage of internal organs. Right here we see part of a colon that is basically bursting with Scaris lumbricoides, this giant intestinal worm. This was in a child, I think like an 18-month-old child who presented with some heavy GI bleeding, and they had to do an emergency surgery. This is what came out, just necrotic tissue in that it's completely obstructed by a very heavy worm burden. Again, like that's what happens to kids. They get very, very heavy worm burdens. So in addition to siphoning off nutrients, that can become potentially fatal if you have blocked or obstructed internal organs. With filaria, the obstruction is more with lymphatic channels. That's what causes that edema and swelling. Cellular damage, Plasmodium, Trypanosoma species can cause cellular damage. This picture in the lower right is showing Trypanosoma cruzi, the American trypanosomiasis organism. And here you see the masculine form that tends to go to the cardiac tissue and causes heart problems, that kind of cellular damage. Invasion of host defenses, we have a couple slides that will talk about that in a little bit more detail. And then finally, these host susceptibility factors. Age is important. Young kids, like we've been talking about, don't have a developed immune system. For that reason, they're particularly susceptible. Immune status is always important. Nutritional status. If you put a tapeworm in somebody like me, who's a well-nourished person, probably gonna be fine. But you put a tapeworm in a young kid who's starving, like they can't afford to have a long-term siphoning off of calories like that. So those can become really dangerous depending on the host nutritional status. And then genetics plays a role in this as well. Yeah.
So there's like excess appetite
With tapeworms specifically? I don't know, like, I'm trying to interpret your question in my head, like you're saying somebody's overweight, do they support tapeworm infections better? Well, I mean, if you have calories to spare, I don't know. I will say that we've co-evolved with tapeworms for thousands of years, and you know how our immune system sort of knows, it has taught itself over time what to handle and what to leave. It leaves tapeworms alone. And not because it's like a diet method, but because it's not doing us a whole lot of damage most of the time. So our immune system usually leaves tapeworms alone if we have these infections. And they have done some weird clinical trials. I think they were stopped with tapeworms from a different species, like pigs. They tried to put it in humans to see if they, I don't know if it's directed for weight loss or control something a little bit more concerning, but that idea has been explored clinically as well through tapeworm treatments. All right, back to immunology, back to buccal and stuff a little bit. So our immune system, I said it was a little hands off with things like tapeworms, but it still will go after even the big worms, little guys and big worms. Let's talk about some of these in a little bit of detail. We know about our innate immunity. We know about our cells that do phagocytosis. For the phagocytes, they will take care of most of those unicellular protozoa, so that's what they're good at doing. with the bigger prey, things like those helmets, tapeworms, things that can be up to 10 meters long, you need a different approach, right? A little phagocyte isn't going to clear a 10-meter long tapeworm in your gut. So what do? The adaptive immunity kicks in. You still have your pattern recognition receptors, your PRRs, right? They're still recognizing AMPs that are specific for parasites. So you're getting cytokine release, IL-5 in particular, and that is driving the special cell that we talked about, and we haven't talked about much since, but back in block 1, you guys remember talking about eosinophils a little bit? So eosinophils are adapted and really, really good for home infections. You guys remember the mechanism by which they work, what their antimicrobial mechanism is for eosinophils? Yeah, and I have a video, maybe we'll watch it at the end, but it shows eosinophils attacking this multicellular worm. It's pretty dramatic. But that's basically what they do. They cover it and they release, they degranulate, they release all of these granzymes that start eroding, degrading the surface of those helmets, effectively killing them over time. Antibodies themselves can also form against parasites. IgG is good, for example, if you have like Plasmodium falciparum or other Plasmodium species. IgG can block them from binding to receptors on erythrocytes. IgA is good for things like Giardia in the gut, secretory IgA. IgE was that subclass of antibodies that we talked about that was specific for immune response, so IgE is important as well. Cell-mediated immunity. CD8 cells, as we know, if it's like a unicellular infection, those will have a direct role. They kill the infected cells. But it's really the CD4 cells at the end that do a lot of that heavy lifting and orchestration, the cytokine release, and the tailoring of the immune response specifically for parasitic infection. A little bit more detail on eosinophils. This is more like a caveat slide, cautioning you on how to interpret eosinophilia or high levels of eosinophils in the blood. A couple things, what they look like. We've talked about this a little bit. Block 1, bilobed, pink cytoplasmic granules, very pink in color when you do like an eosin stain. Fairly small. They have some limited phagocytosis ability. They can release toxic contents of granules. And importantly, usually their numbers are around 1 to 3%. Their counts, 1 to 3% of your leukocytes in peripheral blood. However, points to make, they might be elevated during parasitic infections, but not all parasitic infections. So if you're seeing a patient who you suspect has a parasitic infection, but you don't see eosinophilia in that patient, that doesn't mean you can rule out a parasitic infection. Okay? That's one thing. Eosinophilia does not indicate a specific parasite when it is present. That means you can't use to say, oh, that looks like Giardia, or that looks like trypanosomiasis, or whatever. The extent of eosinophilia doesn't always correlate with the infection severity. So if you have something like an intestinal protozoa doing a lot of damage, like Enteromoeba coli, for example, you might get higher level of eosinophilia, but barely any pathology associated with that parasite. So they don't necessarily correlate either. And then lastly, there are other things that can cause eosinophilia. Cancers can cause higher levels of eosinophils in the peripheral blood. So make sure you kind of appreciate those limitations with interpreting high levels of eosinophils in the blood. All right. Why don't we take a 5-minute break and we'll hear about prosones, and we won't do much more after that, just maybe 5 more slides, but let's get back up at 10:20.
This one, no, like you said the last one. Really, so that med lab is actually other patients. We haven't took it. It's so we could send you to do to sign up and notes.
break. Question for you guys as we're kind of getting settled back down. We talk about the exam, but I'm just curious, did you get pictures on the exam? Did you guys like them? Really helpful or distracting?
It's helpful.
Okay, how many people out there are helpful? The pictures, the images. Okay, a bunch of people. Anyone found them to be distracting or irritating or confusing? Oh, okay. The question was the image wasn't.
Yeah.
Oh, okay, okay.
Yeah, corporates like seeing that helps me out so that it's not real like scalp or feet.
Yeah, okay. Okay, yeah, thanks. I was curious about that. For this one, I haven't looked at the exam yet, like the base exam, but typically Blacklight does have a lot of pictures. But it's kind of like the same approach where if you remove their pictures and go text, it would still penalize you at all, you'd be on the worst for it, so. Okay, thanks, yeah, I was just curious if anyone found that to be problematic or distracting. Alright, so we're driving on with chromosomes, or unicellular eukaryotic parasites. Most of these guys on the second bullet, if it's free living and parasitic, most of the ones we're talking about belong to that latter category. They're parasites. They need you. They need your nutrients. They need your, need you in order to survive. One notable example is the Ignaria fowleri, and that is a free living amoeba, and it doesn't need you at all. When you do acquire Ignaria fowleri, again, classically like being forced up through the curviform plate in your nose and into your brain. It's bad for you, you'll probably die, and it's bad for parasites, also gonna die. Okay, so completely free living, accidental type of thing. Transmission, vector-borne, fecal, oral, talked about that a little bit. Nutrient acquisition, what's the difference between penocytosis and phagocytosis? Yes, so SIP is a good verb to use for penocytosis, just the membrane literally just imaginates a little bit and it takes in a little bit of fluid around it when it does that. That's penocytosis. Phagocytosis, we know, is like a big, it's like the big gulp. And then diffusion is when these nutrients passively diffuse into the cells. On this slide, we have a bunch of different examples of protozoan classes. The guys on this slide that are intestinal protozoans, like, look at my laser pointer back now, Giardia, like this Amoeba histolytic troph here. A lot of these are facultative anaerobes, so they will have specific adaptations to the low oxygen environments of your gut, your colon. Blood parasites as well, endosomes, tissue parasites like Leishmania, Plasmodium. In terms of motility, this is one of the most important things for classification purposes. We have flagellates. So we've talked about flagella before in the context of bacteria that usually might have one or they may be covered in flagella. But with some of these metagonata parasites like Giardia, you have 8 different flagella coming off the body, symmetrically aligned. Some for steering, some probably, most of them are probably for steering and motility. And even Trichomonas vaginalis, you have 4 flagella up front, and then 1 for steering in the back, like little rudder. It's a taxane running down its body. It's terminal flagella. One major ciliated protozoan, this Valentinium coli, it's huge. Not much of a pathogen, but it is a ciliated protozoan. Things like Amoeba histolytica, which move by pseudopodia. They have amoeboid-like movements. That's what these pictures up here, these GIFs are showing you. The movement of pseudopodia and reaching out and grabbing and using whatever they can cling onto for traction and movement. And then others like Plasmodium falciparum, Toxotoxogamia, P-complexa, which means they're mostly non-mobile. If they move at all, it's just by slight flexion of the body. terms of survival, we've talked about cysts a little bit but with protozoans, this is really important for a lot of them in terms of transmission. For cysts, this is a form that facilitates transmission from host to host and allows them to survive. But as a trade-off, this is not the form where they would be doing things like eating or dividing or replicating. Okay, so that cyst form that they can go back and forth between cyst and trophozoite is for transmission and survival mostly. And then with these trophozoite forms, this is a form that would be actively replicating and feeding. That's trophozoite, that's what it means. It means feeding animal. And it is a cycle with, again, Giardia. We use a lot of Giardia as an example here, but it goes through cyst. And then when conditions are good and it can replicate and eat, it becomes a trophozoite, which is more fragile, more delicate. Then it divides through binary fission. That's what you see in the second panel. And then when it needs to be transmitted to you, it will persist. In terms of reproduction, asexual reproduction is the dominant form of reproduction. Most of these protozoans will reproduce by binary fission, which is merogamy. Some of them will undergo multiple rounds of asexual reproduction, which is what we call schizogamy. And a few, like Plasmodium species, will undergo sexual cycles, usually that alternate with asexual cycles, sporogamy or gametogamy, Plasmodium parasites. In terms of pathogenesis, a lot of these parasites don't have a terrible amount of virulence, so they're not intending to kill you. They want to survive on you long term, sort of live in peace with you. It is damaging to you, so they're truly parasitic, but it's still something that we can generally get by with. There are fatal cases, but most of these infections are going to be long term and not incredibly damaging. We sort of covered these factors on a previous slide. Infection does depend on the dose, the host immune status, and whether or not you were getting reinfected repeatedly, so you don't have a fighting chance to heal or recover. Chronic infections are possible, and as we've talked about before, when a host response contributes to symptoms, there's a term that we repeatedly use. Do you guys remember what that term is? Immunopathology. So damage caused by the immune system itself. Host response contributes to things like itching, fever, things like that. Okay. When we start, this I think is in the third deck when we talk about intestinal protozoans. This is a list of some of these protozoans we find in the intestines. By far the most clinically relevant one, well, there should be really two, Giardia intestinalis and Echinopsis fluidica. These two are major intestinal protozoans. The rest of these guys can cause damage and disease, but to varying extents. And some of them are more or less just commensals, like you would have no symptoms at all, just carry them. Blood and tissue protozoans. We have the examples in area and Acanthamoeba. These were the free-living protozoans. T-vag, Trichomonas vaginalis, this is one I mentioned was the only STI parasite. It's the one with the 4 front flagella, the 1 flagella in the back for serum. Plasmodium, Babesia. You guys ever heard of Babesia before? A couple people? So that's big in some parts of the U.S. It's big in the upper Midwest and New England states. Tick-borne parasitic disease. Let's talk about that. Toxo, leash, Trypanosoma cruzi, and Trypanosoma brucei. Alright, so that's an overview of the protozoans, the unicellular parasites. Give you a quick overview of the animals, worms or the ectoparasites. With both of these groups of organisms, we have a huge amount of diversity and variety. There are tons of different types of worms, tons of different types of arthropods, and of those, a relatively small number would be considered ectoparasites that are a nuisance for human beings. Okay, so helminths. These are the worms. We've got very complex life cycles, or relatively complex life cycles. They are multicellular organisms. Wherever they are adapted for parasitic lifestyles. So as you can see, this guy, that looks friendly, doesn't it? Definitely do some damage to those mouth parts. In terms of attachment, they have attachment structure can be categorized or described as hooks or suckers, teeth, or plates, but they all kind of do the same thing. They're used to use attachment and to sometimes extract nutrients from the host, like these cutting plates or teeth. Roundworms, the scientific name for roundworms are nematodes. It literally means round worms. And with flatworms, platyhelminthes, we have two additional groups: trematodes, which are flukes, and cestodes, which are tapeworms. You do need to know the scientific names and the common names for these as well. Okay. So starting early to start associating flukes and trematodes and cestodes with tapeworms, etc. With these helmets, we have many human-specific helmets, some that are zoonotic. We have this image here showing a bear with a bit of a tapeworm issue. You can see that. Bears tend to have some pretty gnarly tapeworm infections. So this is just dragging out behind this bear like that. But infection, and when those worms are dangling like that, they are producing eggs, and those eggs are going into the environment, going into the soil, and contributing to propagation of these cycles. For food uptake for worms, for helminths, most of them will absorb nutrients from the intestinal contents. Most of them can survive better in anaerobic environments. And depending on the life cycle, some of those larval forms might require oxygen. I should have shown a picture of close-up of a worm, but on this slide, in terms of the survival of the cuticle on the outside, most of these guys, if you just look at the trematode and the cestode here, this is the tegument. It's the covering over the body that provides some protection for that parasite. I mentioned some of them do secrete enzymes, like schistosomes, for example, will secrete enzymes that'll just help them make their way through tissues. They move from the liver to biliary ducts, for example, secrete those enzymes. Some of them employ strategies like antigen variation, like Trypanosoma cruzi, where they will they have the genetic capability to switch expression and basically coat themselves with antigens that are not recognized by the immune system. Most helminths are produced by sexual reproduction, so that contributes to that complex life cycle we talked about, and we talked about heteroxynous hosts, definitive hosts, intermediate hosts. The definitive ones are going to be those hosts where the sexual reproduction occurs, not the intermediate hosts. Most of these worms are oviparous, which means they lay eggs. Those eggs have to hatch under the right conditions. A very small number of them are viviparous, live birth. And in terms of pathogenesis, this too, we'll see a lot more of this, but a lot of this is going to be direct tissue damage. Worms like Ascaris lumbricoides, the intestinal worm that we saw obstructing that piece of the colon, those don't really respect anatomic barriers very well. Those worms go where they want, they do what they want, they're big, they can do a lot of internal damage just by moving around. Like they're the size of a pencil, basically, inside the body. So a significant amount of tissue damage can occur as a result of them. I think, yeah, most of these other points we touched upon. Okay, flatworms, very briefly, trematodes, flukes, and cestodes, these tapeworms. The cestodes, this is the first group of parasites we're going to be talking about on Monday, starting with Taeniasolium. I think these are more or less arranged in order. There are different distributions associated with these different tapeworms, so we'll talk about a little bit of the intimacy of them. The spread of most of them is fecal-oral route, and some of them are based on carnivorous life cycles, so eating things like rodents, which humans don't normally do we? But there are ways that we come across and interdict these parasites. Trematodes, the one commonality with flukes is that they all require snails as intermediate hosts. It's very, very important. Distribution for most are in different places, not so much in the United States, although we do have some trematode infections here, but And finally, the roundworms, the nematodes. We've got mighty pinworm, global ubiquitous parasite, the giant intestinal worm, which we do get cases in the United States, but mostly associated with the developing world. Toxicara is one that we won't cover in this course, it's not on the PANS blueprint, so don't worry about Toxicara, even though it's super common and important arguably. Trick, that's the whipworm, that's another intestinal roundworm, causes a lot of damage, anemia. Ancylostomata indicator, those are hookworms, strongyloides, and then these guys at the bottom kind of round out with tissue roundworms, so worms that'll get into tissue and do tissue damage. Oh, sorry, real quick, going back to this. Fecal oral route for a lot of these intestinal roundworms, but when we start talking about tissue roundworms, notice that a lot of them are now spread by vectors. Things like mosquitoes, vectors we haven't talked about yet, biting midges, chrysops flies, simulium black flies, cyclops, that's a tiny little crustacean. So some new vectors to spread these parasites around. And then lastly, this is the last slide, the rest we'll cover on Monday. We'll talk about ectoparasites, index 6. These are usually biting insects, biting arthropods, I should say, sorry, apologies. And as opposed to mosquitoes or sandflies, like arthropods that just land on you, take a blood meal and leave, these are defined as arthropods that need to stay on you, significant latch time, to varying extents. For arthropods like ticks, depending on what species of tick it is, they might stay latched onto you for days, and they continue to gorge and get bigger and bigger before they fall off. But the point is, they have mouth parts that are adapted to stay on you. They're not just biting you and leaving. Some of these guys, like some of the mites that we'll talk about, and some fleas like Shigo fleas, they'll spend their entire life cycle on you. And lice, they'll spend their entire life cycle on humans, so they're very highly adapted to stay on human hosts. Okay, well, that is it. We'll pick up on this slide on Monday. Thank you guys, and good job on that